Okay, chapter 3, video 3 of 3. So focusing on all the stuff that's on the inside of the cell. So first part of our inside of the cell is our cytoskeleton. So this is going to give our cell the structure that it needs in order to do its function. It also allows the cell in order for movement. Our cytoskeleton isn't like our normal skeleton, which is fairly going to stay in the same shape it always does, but with a cell skeleton, it actually can change shape in order for the cell to move, in order for the cell to change shape. So the cytoskeleton can do that as well. So our cytoskeleton is comprised of 3 different structures. We have our microfilaments, intermediate filaments, and microtubules. So going from smaller, medium to large. So this picture shows different cytoskeleton components. So our very large brown tubular structures, those are our microtubules coming off of our centromere here. And then the yellow ones being our intermediate filaments going across. And then around most of your cell, we have these really tiny microfilaments. It's all the way down. There we go. Okay, so microfilaments, tiny ones made of actin. Intermediate, a little bit larger, made of the keratin protein. So giving more shape. This one's just kind of like a web generic structure. Microtubules, even bigger, made of the protein tubulin. And your microtubules can be reconstructed to move different places, create a railroad track system for movement of stuff within inside the cell. So there's some motor proteins that walk along your microtubules so you can send stuff and traffic it where you need to go along the cells. So this is very important when we come to our cell division, when we're creating new cells during mitosis. Helps form that mitotic spindle so we can separate out all of our cell bits to our 2 different sides, so we can split into 2 different cells. Okay, so that's just a fluorescent microscope picture showing all of your cytoskeleton components. This little kind of empty area in the middle, that's where the nucleus is, so there's no cytoskeleton there, so that's why it looks kind of like a gap space. But here we can generally see the structure of the cell based on staining our microfilaments, microtubules, and intermediate fibers. Okay, focusing now on organelles. So these are going to do some kind of specific job inside our cell. So some have membranes around them, like our nucleus, mitochondria, lysosomes, peroxisomes, endoplasmic reticulum, both smooth and rough, as well as our Golgi Complex, which used to be called the Golgi apparatus, if you're that old. Like me. Okay, some don't have ribosomes. Some don't have membranes, and those would be your ribosomes, centrosomes, centrioles, and your basal bodies. Okay, starting off with the nucleus. Big, gigantic organelle in the center of the cell. It stores all of your DNA. So most cells only have one. A few cells, like your red blood cells, don't have a nucleus. They're called anuclear. Or some cells, which we'll talk about in chapter 7, called your osteocytes. Those have multiple nuclei, as well as your skeletal muscle cells also have multiple nuclei. So around it is a membrane called a nuclear envelope. Its job is to traffic what can go in and out of the nucleus. So going into the nucleus, copying a part of that DNA gene, so making our RNA, and then allowing the RNA to go out of the nucleus, keeping our DNA inside unless we are going through mitosis and splitting our cell into a new daughter cell. Okay, so inside our nucleus, our DNA is organized in what's called chromatin, so big, gigantic loops organizing our DNA. In the center portion, we have nucleoli, and that's where our ribosomes are made, so helping us with our scaffolding and creating of our protein chains, which we'll talk about later. Okay, next one, right outside of our nucleus is our endoplasmic reticulum. We have two types. So we have our rough endoplasmic reticulum. Its job is to create phospholipids. So when we're creating a protein that needs to be put into a package, the rough ER creates the phospholipid so we can create that phospholipid bilayer bubble to put that protein in. So proteins that are packaged into a vesicle, they're created on the rough endoplasmic reticulum. The other endoplasmic reticulum is smooth endoplasmic reticulum. So the reason why this one is smooth in surface is because it doesn't have those ribosomes. So those ribosomes on rough ER kind of makes a bumpy surface on the exterior. Smooth ER does not have those. So smooth ER has a completely different job than rough ER, and its job is to create steroids and other lipids. It also works as calcium storage. So later when we talk about skeletal muscle cells, the smooth ER in those muscle cells, its job is to store calcium, so when it's triggered to release that calcium is necessary for the regulation of our muscle contraction. So rough ER and smooth ER functionally different, but they're actually part of the same network. So here we have on the left side, we have rough ER. So again, it's rough because it's studded with those ribosomes. Smooth ER, so it's actually continuous with the rough ER, moving into a more tubular shape of our smooth ER. But again, it's smooth because you don't have the ribosomes. Okay, so what are the ribosomes? So they're a type of nucleic acid. So some protein, a little bit of RNA. Its job is to help read our protein blueprints, so our RNA that we've copied from the DNA, and help to assemble the pattern of amino acids that are coded on that messenger RNA, and help to put these amino acids in the correct order so that you can create your protein. So putting everything in correct order, because if not, your protein's not going to be in the correct shape. Therefore, it's not going to be in the correct structure. Therefore, your protein's not going to work, and it's just a waste of your time. Okay. So with our ribosomes, you have two different types. Some that are what's called membrane-bound ribosomes. These are on the rough endoplasmic reticulum. And then some that are just kind of free-floating, hanging around the cell. So those free ribosomes are for proteins that do not need to be packaged into a vesicle. Okay, different organelle. Golgi complex, also called the Golgi apparatus, used to be. For the Golgi complex, it also creates phospholipids made of a membrane itself. And this is where you get the finishing touches on your protein synthesis, particularly the ones that are packaged into a vesicle. So it kind of sorts your protein around. For example, with hemoglobin. Your hemoglobin has multiple proteins. So in that type of situation, your Golgi is making sure that each of your different types of proteins are being put together correctly. It adds some carbohydrate chains to some stuff. It kind of reorganizes stuff. So sometimes as part of the regulation of protein making, you make more of a amino acid chain that you need and then regulate it later. Some of that part is chopped out and then put together in your final product. Lysosome. So this is a membrane-bound packet with enzymes in it. So sometimes we have excess proteins or excess organelles that we don't need anymore, or if it's a cell that goes through phagocytosis of bacteria, we take those lysosomes, we merge it with that vesicle, or just add something that we need to get rid of, mix it with that lysosome. The enzymes inside the lysosomes is going to chop up and break up whatever has been put together, merged with that lysosome. So we have excess organelles, it's something we don't need. Autophagy is what happens. Or let's say if your cell becomes so damaged that it just goes through your cell suicide called autolysis, then just getting rid of that damaged cell so that you can make room for a new cell. Next type of organelle is a peroxisome, similar to a lysosome, except for instead of completely destroying whatever molecule or organelle that it's bound with, the peroxisomes are just going to create enough damage where the compound or structure of whatever was put in with the peroxisome, it is no longer going to be able to function. So for example, using hydrogen peroxide. So the reason why it does the bubbling is because that hydrogen peroxide, which is an unstable molecule in the first place, it's going to break apart. Those pieces of the hydrogen peroxide are going to find a partner because it wants its valence shell to be happy, and so it's going to find other stuff to attach to, and that causes your other structures to break apart. So this is why we use hydrogen peroxide. Not only our body makes it as part of our immune system, but if you have like a cut or a scrape or something, you put the hydrogen peroxide, it's because you're making sure those little free radicals are able to break down that bacteria. Okay, so this is in all cells, highly abundant in your liver and kidney because you do have a lot of blood filtration there, so getting rid of something that might have infected your blood. Another type of organelles, you have proteosomes. This same situation is to break down something, specifically protein. So if you have a protein that is tagged for destruction, you don't need it anymore, that proteosome will get rid of it. Okay, mitochondria, the powerhouse of the cell. We've heard this over and over again. But really that's what it does. Its job is to take glucose, break it down, make those ATP energy molecules using oxygen as a very efficient method of creating our energy storage with our ATP. So our mitochondria, they are all over the cell. If you look on the bottom right corner, all the green inside those cells are actually mitochondria. So they're all over the cell. They follow the path of your cytoskeleton so that they're able to spread out. Sometimes they can be in the little bean shape that you normally see in your cell structure drawings, but then they also can merge with each other if they need to share nutrients, need to share some kind of resource, and be more efficient. Okay, Centriole, I'm going to skip that one. Okay, a bunch of inclusions. So, of course, you have some pigments or storage of stuff that's also can be hanging out in your cell. You don't actually need it to be stuck in there, but they're there. That it? Oh, that's it. All right. Into chapter 3.